There are previous vision space presentations that chart the story behind why it is that I find myself considering a possible link between dark matter and our sense of vision and perceptual structure in general. A full list of related presentations is on the PAC website under the icon Presentations. Here I want to consider a possible extension to the function of the on-center and off-center receptive fields of cells found in the retina and related parts of the brain. First, it's necessary to examine the conditions that would theoretically give rise to the requirement for the function. This raises a host of questions to which I don't have either the answers or the means to pursue them. Perhaps somebody has. There are well-documented ways in which dark matter makes its presence felt, and there are even suggestions that in rare situations interactions between the matter we can see and dark matter may take place. However, we need only consider its gravitational effects on passing photons, termed gravitational lensing. I need to assume here that the fundamentals of gravitational lensing over cosmic distances are understood. If not, there's a lot of information online. The point being that the trajectory of photons are deflected or attracted by the gravitational force of dark matter as they pass through varying densities of the stuff. Weak lensing effect from less densely populated regions of space can be used as source data to generate 3D maps of where dark matter lies. So the presence of dark matter affects the trajectory of photons over very large distances. But we are also surrounded by dark matter in our terrestrial setting here on Earth. We as sentient beings are embedded within dark matter. It passes right through us and the Earth unnoticed. To what extent can we scale and reapply the understanding gained from the remote observation of light passing across cosmic distances, and through dark matter fields, compiled as though we are a third-party observer stationed outside the system being modelled, to one where we are inside a system, part of it, without recourse to a conceptual box to confine the situation? Does the presence of dark matter affect us at an experiential level, in our everyday lives? This model shows multiple deflections of photons travelling across unimaginably large distances and around multiple clusters of galaxies en route to us. In contrast, we are considering life within a single dark matter system into which we are embedded. Can it really be possible that the deflections dark matter exerts on photons can scale to terrestrial circumstances, discernible as mere microscale noise to biological systems? Obviously, we would presume a smoother photon trajectory than the fluctuating and tortured path illustrated in this model. We would be considering very small deviations leading to effects many orders of magnitude too small for direct perceptual analysis, but nonetheless detectable by retinal receptors. The data then collected through visual process to be deployed in establishing values building to describe aspects of environmental spatial awareness that we do perceive. As photons bounce off objects and travel terrestrial everyday distances through space, with its proportionate volume of dark matter, to what extent does the distance, and hence the quantity of dark matter traversed, affect the recorded relative position of individual photons streaming from the source to our receptors? Can dark matter act on photons as a kind of field or medium over these short distances with respect to the factors involved, like the speed of light? If so, could our physiology gauge the tiny vibrations that would manifest? The dark matter would be being hijacked and used as a medium to describe an environment it actually never encounters. Our receptors do appear to be mediating firing thresholds in response to photon arrival, giving rise to dark light, and this has been linked to dark noise. Do the receptive fields of cells actually receive multiple hits that can be thought of as slightly deflected from a true rectilinear path? Does this outline a mechanism enabling us to think slightly differently about the loss of coherence associated with quantum decoherence? The suggestion would be that a conceptually uniform straight path for all photons from a source would only be possible if dark matter were absent from our universe, or that a mechanical device such as a camera negated the distribution function through the application of hard lenses, effectively dissipating the noise function prior to projection. Of course, the camera is also not designed with the required data collection technology to make the detection, even if the noise function were transmitted to the plate. Could a spatial distribution of hits from the photons streaming into the eye and logged over small areas of the retina establish a disorder value from a noise function embedded in the light array? Are the receptive fields of retinal cells designed to detect and to some extent gauge the noisy patterning to pass forward spatial references? If so, any one spatial reference would only hold meaning when collated with multiple other summations from other receptors or receptor groupings correlated across the retina and collectively expressed as a field function. The suggestion would be that the receptors, gauging the noise potential in the light array, would be detecting the so-called dark light, known to mediate receptor firing. A number of hits would be required across receptive fields of individual and neighbouring cells in order for the threshold value to be reached, involving both horizontal and then vertical transmissions. It would then be essential that the noise element be preserved through and across the retinal layers to influence ganglion cell discharges. Again, this is known to be taking place. Receptive fields have two distinct regions. These areas fire in opposite ways and are known as on-center and off-center. Their functions have been linked to edge detection tasks. Would it also be possible that the off-center cells were responsible for delineating the extent of photon deflection across its surface? This data set extracting a spatial value. The further the extent of photon distribution across the field, the further the reflective surface in the environment would be logged in perceived space. So it would not be edges that are being detected as we consider them in pictures, i.e. differences between light and shade, colour, etc., but spatially encountered edges as they occur within the disorder field. The suggested system would detect significant changes in the spread of hits across or even between receptive fields, these building into a highly efficient, simultaneously understood field, not just identifying the edges of objects, but supplying a homogeneous form of spatial awareness. Spatial awareness would then come before object recognition, and not built from multiple assessments of objective form, followed by the assessment of multiple occlusion cues, and then perspective cues. Spatial awareness is implicit in the data, not deduced from the formation of pictorial cues. Spatial coordinates are embedded in the incoming light array and realized. So spatial awareness is not primarily the result of conceptual analysis. It's not at the end of the line, it's there at the beginning. The conceptual analysis of space is a secondary feature of the visual pathways dealing with what is in the world, and derived as a byproduct of establishing objective form. If the visual system were organised in this way, then the importance of the stimuli presented to the eyes would be absolute in experimental terms. If the intention is to explore visual process, the visual system will require stimuli structured in accordance with its function. We are currently insensitive to this, and the results obtained from experimentation performed with this limitation embedded within the process will lead to compromised results and ultimately a compromised understanding of sensory perception.